Okay, thanks for streaming.
So, I forget the genesis of this. Why we decided to do this? I can't remember how this came about. Uh,
Just as revisiting a lot of basic morphology explains some of the machine learning.
okay. So, they sent out two things. One was both by Vernon Mountcastle, both about his basic theory of cortical columns. One that was this book here, which was originally published in '78, has two essays in it. Sort of unusual sort of publication, but Vernon Mountcastle, like 50-page sort of essay he wrote, and this is where he really, for the first time, laid out carefully the argument for cortical columns. And then there was this other paper which came out later, which is it says a review paper in '97. It doesn't quite read like a review paper. It's more sort of a stronger argument for his original hypothesis, because most of what he reviews, not all, but a lot of what he reviews is his own work. And and then also he points out here somewhere, see if I can find it. Anyway, somewhere in this paper, this one here, he points out, like, well, when I first introduced this idea a long time ago, people made fun of me and they didn't believe it. So here I'm trying to, there's some wording in here. He actually says that. So there's a couple ways we go about this. Mountcastle's writing is always dense. He has a lot of information. It's always dense. And so every paragraph, you could spend parsing every word and every sentence. And so there's a lot. I went through this paper again. I haven't read it in a while, and I have a lot of notes on it. But we could also just do a very high-level organization, just kind of touch the basics of it if you want. And I think maybe given the time constraints, I'll just do that. But there's a lot of detail in here that if you're interested in sort of understanding these systems and how they work, the details matter. And he has some data in here which conflicts with data I've been using recently. So it's a question about numerical data that also is interesting. So let me just, maybe I'll just start with the essay here. And that's this guy here. And how can I do this best? This is like, often when they have these old things, you just got some scan of this. It doesn't read very well. So my book is nicer than this. So I'll walk you through what he basically argues here. In this, in the beginning, whoops, again, it's a PDF, it's hard to highlight things. What he's basically arguing here in the beginning, it's complex language, but he's saying that going back to the mid-19th century, biologists basically argued that the brain evolved over time like species, and that unlike species, as the brain evolved new sections, they were added on top of the old sections, and the brain is sort of a conglomeration of these. He talks about the successive addition of more cephalid parts, meaning towards the head. So you have like the brainstem and the spinal cord, the brainstem and the pons, and then various sort of subcortical structures. And that the argument was made back in the 1800s that as you added new parts on top of the old parts were still there, and the new parts tried to control the old parts. So each new addition is accompanied by elaboration of more complex behavior at the same time imposed the regulation
This
of more
paper has no pictures anyway.
and presumably more primitive behavior. So basically, these things are trying to control the old things.
We still have all the most basic things.
This old things are still there. Yeah, the old things are still there. So that's the basic idea. And then he says, but when it comes to the neocortex, which is 75% of the volume of the human brain, the neocortex got big and elaborate not by this process, but it got big and elaborate by a replication of the same unit over and over again. So this occurred over hundreds of millions of years, whatever. And then, but the neocortex comes along, it's the newest thing on top, and it got big, not because it added new things, it just got big because it went expanded and replicated.
Yeah, tripled in just 5 million years.
Yeah.
Can I ask this question about this old brain, new brain? Do the old parts of the brain resist somehow, like being controlled?
Well, they're in control. So it's, yes, they do resist. So I give multiple examples. This is off the topic of cortical but like you can think there's a lot of old things you do. Take, for example, coughing. That's or breathing. These are things that the old brain does. You don't need a neocortex to do them, but your neocortex can try to control them. And the neocortex can say, oh, I'm going to ask you to stop breathing right now, or I'm going to ask you to do a synthetic cough, or don't cough. But after a while, the old brain goes, screw you, I have to cough, I have to breathe, so I'm taking over again.
Try not sneezing.
Yeah. Well, interesting, sneezing is one thing that the cortex can't control at all. You cannot make yourself sneeze. I can make myself cough. So I can blink my eyes. So there's things that the cortex can control, and there's things the cortex can't control. But the cortex is always just trying. The cortex can try to do these things. Even walking and running are old brain things. You don't need a neocortex to run or walk. But we can, so when you're walking on the street, it's really the old brain doing that stuff, the spinal cord. But the neocortex can try to control it. So, and then he says very shortly after this, let me just get in here. Sorry, I'm not going to spend a lot of time on this book. I'll just read this to you. So he says, sure, I'm just reading on page 9. He goes, I guess I can show you page 9. He says right up here,
Oh yeah, can mark the exact same.
Put shortly, he's basically arguing that the cortex got big by replicating some structure, and therefore the same basic algorithm is going on everywhere. And he said, put shortly, there's nothing intrinsically motor about the motor cortex or sensory about the sensory cortex, meaning they're all working on the same basic principles, and thus the elucidation of the mode of operation of a local modular circuit. If you can understand what's going on anywhere, then you understand what's going on everywhere, generalizing significance. So that's his basic, that's the big idea. And when he proposed this, people laughed at him and basically said, that's not right, it can't be right. It just struck people as wrong, that the same algorithm could be occurring everywhere. This assumption is that language areas of the brain are going to work in different principles and the vision areas of the brain are going to work in different principles elsewhere. So he goes through this in this example here, and he makes, in this essay, he basically makes the argument that the unit of replication, he's a little bit vague about it. At one point, he talks about the mini column as being the unit of replication. That's the one he really focuses on. But he also talks about the larger column, the macro column. So he's basically arguing both, and that and a macro column could be, you can think of like anywhere between 0.5 millimeter, millimeter width, composed of a bunch of mini columns, and that thing, whatever it is the unit of replication. It's just multiplied multiple times. Okay, so that was, it's worth reading this essay. It's not easy to read. Like most of his work is not easy to read, but it's very, very insightful. Then this paper here, I'll just touch on the highlights of this. He's basically making the same argument here, but going through essentially empirical evidence for it. And I'll just jump ahead to show you the basic idea that he is arguing for. Let's see where I want to find the first picture.
Really good figures on the somatosensory.
Yeah. So we're going to scroll down here a bit.
In the first paper, 1917, how did he came up with through like analyzing images?
Well, mini columns are visible.
Visible.
Right? You know, they Cajal saw them. You can see them on the Cajal picture out there, right? So they basically, and we talked recently about the double bouquet cells, and I'll show you a nice picture in here too about the some of the apical dendrites of pyramidal cells are bundled together in these little groupings, and there's a gap, and there's a grouping and a gap. So they're visible in the cortex. I'll just jump to that picture right now. There's a nice picture in here someplace. Here. Just down a bit. So when you look in the cortex and you stain these cells, you'll see these little bundles like this of the axons, gap in between them. And one of the problems is that these all come from, we've talked about this here, they come from a progenitor cell when the cortex is developing. And so people argue, well, it's just an artifact of ontogenesis of the genetics of the cortex. It may not have a functional role, and then they're also not clearly visible in all species of animals. So they're in primates, they're very visible, but for example, in rodents, they're not so visible. And so people argued that this doesn't really do anything. He says no, it's a functional unit, and he's arguing for it. But anyway, this is literally what a mini column looks like. There's about twice as many cells as shown in this particular drawing. So this is about half the number of cells in these layers. And this is looking at striate B1. This is B1 cortex because you see the multiple layer 4s there. But he does make the point that there's a grouping of these acts. These are the tracks of the apical dendrites basically from all these cells, and they're all bundled together. He makes a distinction here, by the way, that in layer 6 there's an exception because in layer 5 and 3 and 2, they all go up towards the surface. Because in layer 6, these apical dendrites terminate in layer 4, which is very consistent with our theory about these being the sort of two sets of place cell-like and grid cell-like neurons in the cortex. So that's the model we've been developing there. So anyway, that's your question there. Did I answer that question sufficient? Yeah. So let's just look at some of these figures. This is the basic method in which they do this. This is like a surface of a monkey's cortex here. This is the actual cortex. And what they do is they stick a probe through the several millimeters of the probe, and they measure cell response properties as they go down. So every little dot here every little thing is like where these are the tracks. That's not a track. That's a distinction between two cortical regions. This is a probe track, and these are little places where they measure the cell activity along that track. So they do these different probes. You see, some of these are very sort of vertical, or they go up and down across the perpendicular to the pedal surface, as you call them. Some of them, if you go at an angle like this, you'll see here you put it in here, but you're actually going across the cortex. So they have to sort of reconstruct these things later to see where did this probe actually go. And what they do basically when they do this, they can see the response properties. Let me go back to bring this up big again. Here they're showing like, as the probe goes down, this is showing like here, look at this one down here. This is showing a part of an animal's limb, and they're showing where as the probe is going down right here. They're saying as the probe goes through these cell layers here, they're essentially, these are all representing the same part of the animal's body at just different size areas. So the argument that these are all actually centered on the same spot, but the different cells in the different layers are representing, they're not exactly the same, but they're all representing the same patch of skin, if you will. More interesting is this figure here, and this gets this figure here is basically showing even in somatosensory cortex, there's these multiple maps of the body. So all these things, this is like going across the surface of the cortex. And they're showing there's like an area here that representing the wrist, and then there's some digits of the fingers. But there's another map right next to it, wrist and digits of the fingers, and another map right. So these multiple maps of the body, they're kind of, it's a little bit tangential thing. Now here's the key figure he wants to make here. He basically, this is the crux of the whole issue. He's saying that if you go across, if you can go really perpendicular and you stay within a mini column, then these are the areas of the animal. This is the limb here. This is the paw. This is the paw of an animal here. You see that? These are claw and claw and claw. So these cells that he's recording from here, they all represent the same area on the pad of the animal's paw. And so they just go straight down, and you're staying within the column, I should say, within the column, the macro column. Here's one that goes at a slight angle. And what he says is what happens, you go to the slight angle, they're first of all representing the same area, then it jumps over here and now represents an adjacent area.
And that's discrete.
That's discrete. This is the key item there, right? That as you go across the surface of the cortex, you don't see in the anatomy, you do not see physically distinct breaks. You can see the mini columns, but there's no other anatomical distinction between as you go across. But physiologically, you do see this distinction, at least what the cell is representing. There's a discrete jump between one column, and then the next column is representing an adjacent area. It's not like, it's not like our two. So in our theory, where each cortical column is doing a complete sensory motor inference, we rely on that property. All the cells in that macro column and the larger column are basically building a model from the input from a patch of sensory area, and then the next column we're building a model from the next patch of sensory area. If this wasn't true, our job would be much harder, but he's making this point. And then he shows, here's another example where, again, it's as you move the probe across these different columns. So this is a macro column, a macro column, and a macro column. As you move across these macro columns, you see it jumping from once, this is a cell, bumps over here, cell, jumps over here, cell, jumps over here, cell. So this is the crux of his argument, that there is this column organization, even though you don't see it. It's important. And then he goes through, he basically says, okay, his big thing was somatosensory cortex. That's what he spent most of his time. So that's what this paper begins. And then he says, how about the visual cortex? And then he goes back to rely on various different types of things, even this metabolic and blood flow studies, which they don't do anymore, but it was interesting. He basically argues that Hubel and Wiesel found the same sort of thing in visual cortex. I won't go through it unless you want to. And then he goes you might have seen these maps before. These are how different properties are laid on top of the cortex. But I'm going to stick to his basic argument for a moment. Auditory cortex is the same thing. This is the mapping in auditory cortex, and he makes the same argument that in auditory cortex you have the same sort of column jumps to a new property, column jumps to a new property. But in this case, it's looking at these frequency bands. And so a column of cortex will be looking at a band of frequencies, and the next column over, we're looking at a discrete different band of cortex. He also makes the point that within a macro column, the individual mini columns are not ordered. They can represent different things, but they're not ordered. It's not like within the column, there is no continual movement across the surface of the. So the mini column at one edge of the macro column and the mini column at the other edge are not like further apart. So he basically says this column organization exists in somatosensory cortex and auditory cortex and visual cortex. There he goes. And then he talks about motor cortex, specifically breaking that out from somatosensory cortex. And makes the same argument there. And then he talks about columnar organization in homotypical cortex.
So what is in motor cortex? Different columns are controlling different.
Yes. Yeah. I have to go look it up. His writing again is very dense. It's hard to keep it in mind all the time.
To project a single pool of motor neurons.
Yeah, but it's also it was more complex than that. So I'm going to read what he says right here. It says the layer 5 neurons, which are the ones that project to spinal cord, clustered into groups, distributed intermittently in horizontal dimensions. So he's saying that the layer 5 cells are grouped as you would expect in mini columns. And then you say clustered into column aggregates 300 microns wide, so that's a macro column. He says 40% of the neurons in each cluster project to a single motor neuron pool in the spinal cord. The remainder project to motor neuron pools and muscle groups active in similar movements. So he's saying that these cells all project to a sort of pool of neurons that are all related in the spinal cord. It's not like, again, it's sort of a segregation. And I followed some of these references. It's very complex, this stuff here. They're making a distinction between motor neuron pools and different these behavioral groups. But he's just trying to make the argument that it's the same all the way down. Here's something that's interesting you might find interesting here. I have lots of notes on this. So I just didn't think we're going to go. They don't have time to go through all this. Here we go. Here. I don't know why it's doing the one. I only want to do the ones on the right. All right, here we go. The question of relation of muscles to movements in the motor cortex. He said basically how the motor cortex projects to the spinal cord plexus is a sort of unsolved mystery. The question was misdirected, but the problem appears to be not whether the muscles and movements are represented per se. So they're saying like, oh, does a motor neuron in the cortex control a muscle? or is it control on movement? That was the question that people were trying to solve. And he says that wasn't seem to be the right way of asking the question. He says how sets of neurons related to particular muscles are combined in groups that from time to time have different compositions and thus generate a variety of different movements in which any muscle participates. He's basically describing SDRs.
Yeah.
So that's how we think about it too. It's always a sparse population that the same neurons can do different things. So you can't look at a motor neuron and say, oh, it controls this muscle. And you can't look at a motor neuron and says it controls this behavior. You have to look at a population code of motor neurons that then can encode a particular behavior. And the same neurons in a different population code control different behaviors. So that's, yeah. There's so many nice things in here. I mean, it would take hours to go through all of this. Here's a funny one over here. I wrote ha. So now we're looking at this. We're looking at this side here. He's saying, so now when he talks about homotypical cortical areas, this is what people sometimes would call association areas. These are areas that are not clearly a particular sensory modality. All right. So other parts of the cortex, it's not so obvious what they're doing. That's what he's calling them homotypical. These properties are complex and rarely ever do the input linked properties of defined modules and sensory areas appear unchanged in homotypical areas. So he's basically saying it's not easy to figure out what these things represent. They seem to represent lots of different things. But then he says here, the plasticity of cortical synapses suggests that homotypical defining properties may be changed by experience. For example, with the learning and memory, and there's preliminary evidence that this is indeed the case. The reason I said ha is because we think that all cortex is learning all the time continuously, but back at this time, that was viewed as a radical idea. And that most of these cortical regions are sort of set and doing their things. And saying there's evidence that actually there might be plasticity in these higher regions. So we might learn something here, which is like, fun.
This idea of a fixed cortex still pervades so much of neuroscience. It's quite annoying.
Quite helpful also from the Tom was the early part with the crushed hand. No, that was not in this paper. Was that in the original essay?
The crushed hand? I don't think
the crust, the resutured.
I think it's in this
that was in this paper. That was in this one here. Right here, this one.
Yeah, that's a bit useful to understand. So if you look at cortex, because the way that these axons originally grow, right, the neighboring columns process neighboring pieces on the hand. So they're actually neighboring the cortex. But when you delete the nerve and then attach it back together, it turns out the whole thing reorders.
They reconnect. The nerves
right?
reconnect.
So the cortex does not tolerate empty columns. They have to do something. And of course they're going to be in the same ballpark, but it's not the exact same order again. As you can see, it's not like crossed over and it's slightly shifted on the hand. So the point being that one should not be fooled from the mapping to think that it's necessary, that like all the things are distributed and they're discrete. And
yeah, this is again the argument that the column is its own kingdom in some sense, right? And so it's not relying on this continuousness of the sheet. It's like saying, hey, fine, I'll mix it up.
Right.
It's no problem, and I'll learn to deal with that.
I mean, like some adjacency is probably necessary, right? For the this digit and this digit, they share like a little bit, so they can't be crazy far apart. But they don't need to be neighboring necessarily. Like the lateral connections are long enough that you get your signal somehow.
Well, in our theory, the columns all work independently, but they do share a couple of things, right? They have to share through our voting mechanism. They have to share what objects they're looking at. We've also talked about how they have to share their relative positions. We've talked about a lot. Marcus will remember that. Some of that can be long distance, some of it's going to be local. Probably the sharing of the relative positions is going to be local, so you couldn't have these things separate on different parts of the brain. But but actually you could, because there's these, if you go from the left hand to the right hand, there's these very strong connections between across the two hemispheres.
And exceptions to exist.
Yeah,
There's certainly places that are supposedly far apart are actually close apart because it is a direct fiber.
Yeah.
So
But we have theories for why that all is. So he's still just trying to make the argument, and as I point out, people still don't believe that these columns really do exist. They are, even if you don't see them anatomically clear, that they are there. And
there's a tremendous amount of confusion about these. Even yesterday in our conversation, the person said, I won't mention the name, but she said, "Oh, you only see columns in the barrel cortex," and that's because they're physically visible there.
Yes.
But this is a functional definition, not a physical anatomical definition.
Yeah.
And so people confuse that a lot.
Also, the distinguishing between hypercolumns and minicolumns has not helped that understanding.
Yeah.
The fact that you're arguing about nested modulatory units
Yeah.
doesn't help people comprehend.
People confuse and people use the word column.
And I think Mountcastle wasn't. I mean, I love his writing. I've learned so much from him, but he was not as clear as he could be here, because sometimes he uses the word module, sometimes he uses the word column. I even caught a few places here where I said, I think he used the wrong column. I think he meant mini column here, and he said column. And I'm not certain, but it sure reads that way. So I can see why it's really confusing. He wasn't very consistent all the time about here's the language, here's exactly what I mean. And when he tries to be consistent, he uses language again, which is difficult, because that's his nature of writing. He's a very sort of precise, dense writing. Anyway, I'm if I could go through every page here and pull out interesting stuff, and I'm happy to do that. But
is there anything new you learned from him?
A couple things. I felt if we really want to understand essentially how motor output maps to behaviors, that there's a tremendous amount of data that's available that we're not familiar with. And so he was going into levels of description. And I brought this book along too, because he gave me this book when I saw him 10 years ago. And this is all about the hand. And this entire book is about the hand. And it's beautiful, but there's so much data here that if we wanted to get into that, I'm saying there's a lot to be gotten into there. This is in the same series as this book, which is one of my favorite books of all time, which is about the cerebral cortex. These are just beautiful books. And they have just tremendous amount of data. And I've always recommended this as a place to start if you want to learn about the neocortex. The other thing, it's interesting here. One thing that I think we just need to be aware of, going right back to the beginning here, he starts with some data, which is kind of odd. Now, again, remember, we're looking here at 1997. So this is 20 years ago. He said the sheet surface area for human neocortex is 2,600 square centimeters. I've been using 1,500 square centimeters. That's a pretty big difference. He says the thickness 3 to 4 millimeters. I heard that's only in V1, but I've heard it's like 2.5 millimeters elsewhere. So what's right? This number we know is wrong. Contains 28 billion neurons. The latest estimates are around 18 billion neurons. So, and then he says here the number of synapses on the order of 10 to the 12. Which, if you take that being the number of neurons at 28 times 10 to the 9th, that seems too low.
Yeah,
so
800 synapses.
Yeah, so I like what's going on there? That doesn't make any sense. So already I was walking in here going, wait, some of these numbers just don't add up. What the hell am I supposed to make of that?
Also, science has advanced, right? I mean, we haven't had really even a good full brain cell count until rather recently.
Very recently. But I would have thought that he knew that number of synapses per neuron wasn't right. Even back then. Anyway.
Is this number the same as we use for minicolumns?
Well, I think that the number people say now is more typically, the number that's bantered around more commonly now is 110. Okay. But there is an exception. The one exception that in certain primates with the striate V1, it's twice as much. So in V1 primates, not all mammals, but in certain primates, there's these extra layers in V1. And when you add those in, it's twice as big and thicker. But I think the 80 to 100 is shouting distance. He also points out, and now even here, he makes the argument in this paper that most of these columns are on the order of a 0.5 millimeter wide, anywhere from 300 to 600 microns. And but in the CAT V1, if you go back to Hubel and Wiesel, that's more like a millimeter. So, but all the evidence he gave was closer to a 0.5 millimeter. That's a pretty big difference. So it makes me question a little bit in our models where we are saying, okay, each cortical column is a complete sensory motor learning system. Each one has to have a reference frame. To implement that reference frame, you have cortical, you have grid cell-like cells in each cortical column, and now there's evidence for that. But as these columns get smaller, it gets harder and harder to have enough cells to do the full grid cell and place cell things. And so it's possible in my mind that we have to be open to the possibility that the grid cell substrate that's we believe in throughout the cortex may actually, it could potentially be not constrained to a cortical column. It's nice to think of it being constrained to one, but maybe not. As these get smaller and smaller, the number of cells you have to work with gets smaller and smaller. So then it just forces you to maybe rethink things a little bit. Maybe columns, he argues that macro columns inhibit each other. He didn't give a lot of evidence for that, but he argues that. And so it's possible also that you might have two macro columns that are essentially competing with each other until we divide up the sensory motor modeling system to two or more groups. It's just the numbers get smaller. If we get down below a 0.5 millimeter per cortical column, then I get a little worried about the numerical issues that we're dealing with. So it's on the edge in my mind. So that's something I learned here. And I think that today people are saying that, but he had a lot of data about that.
So if these are computational units and the 3/8 cortex is 2 times larger, does it mean it's computing something harder?
We don't know.
What is it associated to?
So first of all, let's bear in mind that not all mammals have this. All right. So I believe dogs don't have a striate cortex. So I think
it's tied to whether you have color vision.
Oh, is that right? I didn't know that was tied to that.
And also stereo.
Stereo, yeah.
Is it just additional bandwidth?
Yeah.
So vision itself does not require this. We just remember that. So now we can think about striate cortex as an additional enhancement, an improvement upon the basic mechanism. And we have, of course, in all throughout cortex, and I don't think they knew this back then, but in visual cortex, in somatosensory cortex, and auditory cortex, there are these parallel pathways, the what and no pathways. And so in visual cortex, the retina projects to V1, and it has these extra, they call them extra layer 4s, but we really don't know. They're just extra input layers. And then it divides from there. And one path goes to the ventral pathway, one goes to the dorsal pathway, so you have your what and your where pathways. And so it's almost like those two are combined at the beginning, and it's like, so there may be some kind of cleverness going on there about combining, being more efficient at locating things and knowing what they are early on in the visual system. But the way I kind of view it is, since we know that vision does not require this in an absolute sense, we didn't have to deal with it in our theories. We want to understand the basic, take somatosensory cortex, or take, auditory cortex, or take non-primate visual systems and say, okay, the mouse has a visual system. It basically has one cortical region, V1. V2 and V4, whatever, are these little teeny things.
Keep in mind that the mouse is practically blind.
What's that?
Keep in mind that the mouse is practically
blind. Oh, they see pretty well.
Wow.
I used to think that, but then it was Tony Zadora showed us these videos. These mouses, they see things really well. So you've seen these videos? They're really amazing. There's these little pieces of food in front of the mouse, and they're so odd little shaped foods. And the mouse takes its paw out there and grabs it extremely deftly. It knows exactly how to grab that little object in the right position and bring it up to its mouth and nibble it at the right position. It's not like, it's like if it's oriented differently, he reaches around, grabs a little differently, brings it up. So that is purely vision. I was blown away by this. It's like you're looking at this and say, that's like a human. it's incredibly clever.
You see the object and you already know how
He sees the object and he knows what part to eat in that and how to grab it in a way to bring that little part up, and then nibbles at it a little bit and looks at it, nibbles a little bit. So it's not a very great visual system, but it's not like the device is blind. I think clearly it's now seeing the structure of that object and knows how to grab it in detail. So I was blown away by that when I saw that. Anyway, but regardless, obviously the mouse's visual system is not very teeny little cortex, right? So it can't be that great. But the point is in the mouse system, you really have to expect that the mouse, almost everything he does is in V1. V1 occupies a large portion of the cortex, the entire cortex of the mouse, and the higher level regions are just tiny. And so V1, basically, the mouse sees only using V1. And that's pretty much the end of it. Now, we can explain that.
There's another interesting thing of data is that mouse's retina has more sophisticated feature detectors than primate retina. So ours are very basic kind of on-off detectors and things, but the mouse's retina actually detects oriented edges and stuff like that.
One of the things I
like the video. There's only 5 likes. There's like 16 viewers. Please.
Prey tends to have a visual field that goes out to the side for predators, whereas in primates and other predators, it tends to look forward. So stereo vision is going to be more enhanced with the predators, whereas the prey is going to be more of an alert system to say, is there something that's, not quite copacetic about, what's about to creep up on me type of thing. So
eye placement tells you.
Yeah, right.
You're a top predator.
So I'm just wondering if when you're looking at these various brain structures, whether they evolved to support those different roles of basically global monitoring versus targeted, activity.
So thanks.
Well, do you mind if I share your email with in case someone wants to ask a question?
Yes,
Please. Thank you.
Well, the structure of the cortical column is similar regardless of whether you're predator, right?
Right, but you have striate and various other.
I think the point is that cortical columns really don't know what the hell their input means. They just don't. They basically are discovering structure through a sensory motor movement mapping system, and it could be the things. See it by. And so the right way to think about it, and Mountcastle's argument is that they don't know what their input means. They're not designed to do anything particular. He acknowledges, and we do too, that there are variations between different regions of the cortex. So people have argued against our algorithm for that. But the variations are small compared to the commonality. So you got like 90% of the circuitry or 95% of circuitry every cortical is basically the same. And you got small variations here and there. So the basic idea is, what is the common algorithm? I just hear some other things here I highlight here. Here she's talking about the size of columns vary between 300 and 600 microns. And that's consistent across species whose brain volume by.
That's shocking, actually. Right? That doesn't scale at all.
So
The size. Everything else about us changes with size.
Yeah.
Like the heart and the ears and the eyes and like everything. Like if you have a human being that is half the size, everything's gonna be half the size smaller, right? But like these smallest units of computation, they stay the same.
Yeah. So that was his base. That's one of his arguments.
They are all mammals, right? You're talking about
mammals. Only mammals having a cortex. But here's an interesting one. The primitive cortex of reptile is generally similar. Is generated similarly, although the laminar and columnar organization is less pronounced. Now, we've talked about this. We've gone through some papers here about non-mammal brain regions, but he says it's like. So I'm just surprised he said this. It's not inconsistent with what we read, but he makes it sound like, oh, reptiles have their own cortex. Well, they have these various types of older structures that are laminar and so on, but. It's interesting to ask, even in those older structures, there might be the same sort of reptilic things going on as in our brains. And then, of course, this is another big theme of the cortex. Several variables can be mapped onto the two-dimensional surface of the neocortex. This is clear in the primary sensory receiving areas of the brain and appears likely to be so for all areas in which systematic maps have been observed. This is this basic idea that within the cortical column, across the sheet of cortex, you have these multiple maps that are being laid down. So a cortical column is getting some intersection of those maps. And so the maps could be color and or not color, or it could be binocular or monocular vision. Some columns are just going to deal with binocular vision. Some columns are going to deal with monocular vision, and so on. So essentially, and then there's some areas like in somatosensory cortex, there's all these different modalities that are laid on top of one. And
multimodal, like receptive cells.
Yes, but
they respond to one modality, but mixes of them.
Yes, but they're all, in some sense, he's saying they're all representing the same some such physical place on the sensory array. So you're not going to get color from over here and motion from over here. It's going to be color and motion and dark and light, or you're going to have deep skin receptors and vibration receptors and things like that on top of it, but all from the same part of the. You don't see you don't see mix in a column. You don't see input from a hand and another one from the tip and another one from your knee. You don't see that. Right. Within that area, there's multiple modalities. The skin has many different sensors. Your eyes have different sensors, and so they just basically map them on top of that. So anyway, I can look at. I just keep going here, but I think we probably run our course here. There's a slide. I gotta. Do you mind if I take this? I'll call you right back shortly. Good message. A good portion of this paper you can skip, my opinion, and that's starting here. The ontogenesis of the cortex.
Yeah, that's what I said in the email. That we can skip that whole section.
Ontogenesis is how structure changes through in utero development. So how
I found that cool, but it's total side knowledge.
Yeah, it's cool, but it's not necessary. Have you ever heard the phrase ontogenesis recapitulates phylogeny? Ontogeny recapitulates phylogeny. Well, that's a discredited theory, but where the idea is that if you looked at how the brain grows in utero, it recapitulates how the brain evolved over millions of years. So, simple structures, anatomical structures, so on.
I was actually looking for that, but there is this weird discrepancy, right? So, like the mechanism, it involves building the cortical layers like inside out. So you start with the lowest layers, 5, 6, and then you build stellate cells, and then the next cells that get built out with lots of pyramidal cells, and then the next cells that get built get small pyramidal cells for layer 2. And so they get inserted in this column, right? So there's these producing progenitor cells that produce all of them. That's by their genetic clones.
And then they follow this little
and they follow like the glial cell that like essentially sends like it's like you can think of it like a rope. That all the cells are climbing up to insert themselves into this column. This is a very nice figure actually, which explains much.
Are you in favor of that theory?
No, this doesn't tell you. It's not really telling you anything functional.
Right.
It's just,
the weird thing is just that the early cortex is just the superior layers. So like in terms of evolution, like layer 2, 3 came first with an input layer 4. And then we later on added on, presumably when cortex got large, the infragranular layers. And so, but that is not how it gets created. Actually, in humans, we populate the lower layers first.
This is really just an argument. He's just saying, here's stuff I've learned. How I've learned. Here's what we've learned how the cortex grows. And so it's interesting. But in the end, you can just accept that these mini columns are there, and it doesn't really matter how they got there.
Yeah, I found it, the fact that this whole section is right in the beginning of the paper is a little bit of a thing to deter people.
Yeah, and
that's why in my email I said just.
It's the same way in his book here. He's an encyclopedic knowledge about everything cortex, and he wants to tell you it all. So you pick at each chapter and say, do I want to read this or not? You know, I read it once the first time. So that's when you first you hear the radial. Where is that? The radial unit hypothesis, and you know, he's basically saying that's what Florian was just saying. It's like, you know, these units follow this radii of the glial cells, and that's like, you know, fine, it's really interesting, but it doesn't really tell you what's going on there.
What kind of experiment can you do to track how the neurocortex grows? Like, do you have like monitor same brain growing?
What's the first thing to do? They take an animal at different stages of gestation.
Okay.
You know,
Not the same animal, just.
well, no, obviously not the same animal, but the same species, right? So you take a mouse brain, you look at it 2 days old, 5 days old, 10 days old, 50 days, whatever, and you look at you tease it apart.
Including like prenatal development, right?
That's what I'm talking about.
Yeah.
The mouse is pregnant, and they take out the baby, and then they take a look at what it looks like.
There's gonna be someone who like decapitates the mouse babies.
Yeah.
The ontology philology argument, I think, came for not so much for the brain, which if it's disproved, but the fact that the structure seems to recapitulate evolution. When you look at the embryo growing, that there's a tail that disappears.
Yeah. Looks like a fish at first.
Yeah.
Right?
But they also apply it to the brain, so that was.
No, it holds true for a lot of things. It's just not strictly true.
Yeah.
And occasionally evolution finds ways to, you know, not have to recapitulate the entire thing that it did before, but find shortcuts. Just people over parts that it's going to delete anyways.
Yeah, well, there's probably good reasons for trying to shorten the gestation time by not repeating their useless steps.
Yeah, exactly. Right. Well, why generate a tail if you're just going to delete it then, right?
Here's this this right. Let me find it. This guy says here. I mentioned this earlier. He's talking about the mini columns, right? And the mini columns, 50 to 60 microns in diameter. It is discrete and homogeneous. The receptive, meaning discrete, column is discrete, homogeneous means they all look the same. The receptive fields of neurons in mini columns of a column, of a macro column, shift about the columnar focus without topographic progression. That was that idea that a mini column might represent some part of it may represent actually some subset of the area of the input space, but it's not topographic, meaning as you move across the column, it's not like there's a progression. It's just each mini column seems to be doing its own thing. So we can understand that if the mini column is the output of the spatial pooler, the spatial pooler, for whatever it wants, the reason, could say, I want to find a pattern. If given this input space, meaning this area of the sensory ray, I might want to pick out a pattern that's only over here, and another one might pick a pattern over here, but there isn't a progression of that. So that's consistent with our spatial pooler model. You know, you have to really read into all these things. Here's another one that's interesting. I think we've talked about some of this, and then I know it's lunchtime. He's talking about somatosensory areas again. And just on the left side here, these 3B and 1 are somatosensory areas. And they say these areas, these columns in these areas, receive input from both the rapidly and slowly adapting sets of cutaneous afferents. So what that means is cutaneous means like the skin, I think, and afferents are just the inputs. So from a patch of skin, there are rapid and slowly adapting sensors. Like heat would be, or pain is a slowly adapting sensor. I don't know which ones he's referring to here, but adapting, meaning how quickly they come and go. And then he says, area 2 receives inputs largely from joint afferents. I thought this was interesting. Although it also contains complete representation of cutaneous or skin surface of the hand. So, so this is the idea we were talking. It goes back to stuff we've talked about in the past about the proprioceptive system. Mm-hmm. So an individual column could be getting input from both like something with its information about the joint and the skin that's associated with that joint. It's kind of, it was a little bit interesting. That's a little kind of, again, it's almost like a little bit different than the idea that it's all representing the same part of the skin, but saying, yes, it's part of the skin and the joint that actually moves that part of the skin. So that was like, oh, it's like a little clue, you know, these are like, there's lots of little clues here. Anyway.
Is area the same as region?
Area is the same as region. Yeah, region. All right. I probably have to speak for them. Can I do that?
Okay, should we pause?
Yeah. I'll be happy to continue talking about this over lunch again.
All right. Yeah, please.
Okay, everybody, thank you for joining the live stream, and please remember to like the video and subscribe to our YouTube channel if you want to see more content. I try and live stream as much as I can, so subscribe to the channel to see more content like this, and have a wonderful weekend. Bye, Mark Brown. Bye, Bellio. Bye, Falco. Bye, Charles. Who else was in here? Rick. Bye, Rick, if you're still there. Cody. I think I got everybody. B. Dimitri. Thanks everybody for watching. Take care. Have a wonderful week.